Hello everyone, I'm Mosab Awadelkareem from Sudanese Researchers Foundation. It is my pleasure to welcome you to Sudanese Researchers Foundation webinar series. I will be the moderator of this webinar, and I'm here with Rania Baleela from University of Khartoum, and Mushal Allam, who is from National Health Laboratory Service in Johannesburg, South Africa, and with J H Elliott, who is our presenter. I want to thank you all for joining us. We are just about to begin our webinar on Drosophila as a model for human diseases. In this webinar, participants are going to learn how flies model human diseases, how flies are used in drug development, and how flies are used to determine the cause of the disease, and also how flies are used to construct genetic pathways of related mutation. And at the end of this webinar, we will have question and answer session. So for our online viewers, if you have any question, please post it on the event page on Facebook. Now, and without further ado, let me briefly introduce Chris. At the Department of Biology, University of York in the UK, and his current research focuses on neurodegenerative diseases, including epilepsy, dystrophy, and Parkinson's disease. He's particularly interested in the physiology of fly models of Parkinson's disease, and over the last 10 years, he has focused on the role of amines in fruit flies, developing new techniques to find out how they work and how the manipulation of dopamine and Parkinson-related genes affects them. Most recently, his lab has been testing novel drugs related to Parkinson's to see how they can prevent or slow the progression of the degeneration of the CNS. It is certainly a pleasure to have Chris presenting this webinar. Welcome, Chris, and we are so glad to have you with us. I know that you have a lot of information to cover, so I'm going to turn it over to you at this time. Chris?
Okay, thank you very much, and I hope the technical side will run smoothly for the whole time without any interruptions or flaws. But please, if there's something that you don't understand or the link goes, please let me know and we can talk about it. So today I want to talk about the role of flies in the analysis of Parkinson's disease. And we'll start by just reminding ourselves about Parkinson's disease. And then we'll talk about particularly the work that's been done with Pink1 and Parkin. So that's done mostly in other labs. And then at the end of it, we'll talk about some of the work that I've been doing and seeing how it goes. So Parkinson's disease is really about the loss of dopamine neurons in the substantia nigra. And so if you don't have the dopamine neurons in the substantia nigra, then your movements are severely affected. So the movements depend on a circular link between the cortex and the thalamus, and the substantia nigra neurons interrupt this and control its activity. And so the most prominent and most difficult problem in Parkinson's is the difficulty of starting and stopping movement, of resting tremor. The muscles contract and don't really quite know what to do with themselves. And the dopamine neurons tend to play a role there, but we've also got dopamine neurons in other places, in the retina, for example. So people also have problems with eyesight, and dopamine neurons in the gut, and people have problems with things like constipation. So there's a whole range of different symptoms that are down to the loss of these dopamine neurons in different parts. But the one that's most clinically important, one that gets people to go and see the doctor, is usually the problems of the loss of the dopamine neurons. So in the top right, you can see what's happened if you cut a section of somebody's brain through just behind their eyes. You find the substantia nigra, and it's called the substantia nigra because it's full of dark black substance. Substantia nigra just being the Latin word for black substance. And in Parkinson's disease, then you get the loss of these neurons, so you don't get the black staining. And so until relatively recently, the key diagnostics for Parkinson's disease was post-mortem examination of the brain. And it wasn't very easy to distinguish Parkinson's disease from other things until you went to autopsy. There were many similar diseases like multiple systems atrophy that were problems. So if we're going to understand what happens in flies, what we would like is for the flies to make a good model organism, so they would show the same problems. So the idea is that the fly has the same sorts of cells. It may look different on the outside, but inside it's doing the same basic cell biology. And so you might hope that if you have a loss of dopamine, then you would get the same effects. And indeed, if you make dopamine deficient flies by knocking out tyrosine hydroxylase, which is the gene that you need to make dopamine, then they have locomotor deficits, they have difficulties with activity of one sort or another, they have difficulties in feeding, and it makes for quite aberrant behaviors, which in some ways resemble the symptoms in people. And about 15 years ago now, there was a complete revolution in Parkinson's, because doctors had always been taught that Parkinson's disease was something that was entirely environmental. And suddenly somebody started to discover that actually there were genes linked to Parkinson's. They're very rare, and they only account for a very small proportion of the population, but they're there, and they've been identified. So the first one to be identified was alpha-synuclein, which gives you Parkinson's symptoms in your 30s, and then subsequently Parkin and Pink1, which give you symptoms in your 20s. So these are young onset diseases. They happen much earlier than typical Parkinson's, which is produced by late onset, 60-year-old people or more. And so most Parkinson's is still of unknown cause, some 90% of it. But we've got a lot of interest in the genetics of this, because one or two very famous people have had Parkinson's disease, particularly Sergey Brin, who was the head of Google. And I know we're using Google products to do this, so Google is making a fantastic contribution to our lives, making a fantastic amount of money out of us. And so at least some of it is going back into Parkinson's research, because Sergey Brin had his genome sequence, and he knows that he's got the LRRK2 mutation. So having discovered that we can having discovered that people have genetic Parkinson's, you can say, what about the same genes in the flies? About 70 or 80% of the genes are found both in flies and in humans. And you can come along and you can attack those genes in a variety of different ways. So the traditional ways were to X-ray flies and look for changes. That tended to delete large sections of the chromosome. Or you could use a chemical EMS or ENU, which are chemical warfare agents, and they tend to produce point mutations in the DNA. So if somebody's doing that, they wrap up well in the lab to make sure that they're protected from the harmful effects they're subjecting the flies to. And then more recently, people have used a scheme called the GAL4 UAS system to create transgenic flies, either to increase the expression of genes or to reduce the expression of genes by targeted RNAi lines. And in the last 5 or 6 years, people have started working with CRISPR and Cas9 to do targeted mutagenesis. And we had to go at it, and some of our flies have worked, some haven't. And again, as with many of these techniques, you need to do your genetics really cleanly in order to ensure that you hit the gene that you're interested in and not some other gene. So the most widespread of all these technologies is called the GAL4 UAS system, where you have flies in which a particular promoter is driving GAL4 expression. So GAL4 is a yeast gene, not ever found in flies, and it's been hooked up to a particular gene in the fruit fly, to its promoter, so that it produces GAL4. And in the fly shown on the left, it's got the gene sitting in a particular enhancer trap so that the GAL4 is being made. And in the fly on the right, we have a different part of this system. We have the UAS system to which GAL4 would like to bind. And then next to the UAS system is your gene of interest. For example, if you wanted to produce a fly with extra Parkin, you would put the Parkin gene next to the UAS sequence. So you can make these sorts of flies, and then you cross them, and you get lots of offspring with, and in the offspring they have both the GAL4 and the UAS, and so the offspring produce Parkin, but the parents don't produce anything. So it's a nice clean system that you can test for controls, what the parents are up to, and you can test what the offspring are up to and see if they're really different. Or if you want to take a different gene, then you could express Pink1. For example, you make a UAS Pink1 and you cross it to the enhancer trap, and you get the flies being produced as you want. So a whole range of these have been made, and many of them are available from our friends around Europe and in the United States. Though we've noticed in the last couple of years it's got much more difficult to transfer flies across the Atlantic in either direction. And particularly what's interesting is that the GAL4 UAS system gives you very targeted expression of what's going on, so that you can put the gene that you're interested in either into every tissue in the fly or into specific subsets, for example, into the eyes or into the dopamine neurons. So how do you make these sorts of transgenics? Standard molecular biology says that you take a UAS vector and you land your gene into it. So in the black bit on the right-hand side, your gene gets landed. This one will do about 7 or 8 copies of your gene landed into it. And you land it into the fly genome at a particular site, ATTB. So there's only one place this vector is going to land. And so you inject your vector into the eggs of white-eyed flies, and you look for red-eyed flies. So in addition to your gene, there's a mini white that's up at the top. And so every fly which has successful injection will have red or orange eyed flies. And so you get your gene, and you can detect the flies which have been successfully injected and throw away the flies that haven't been. And once you've created the flies, then you can measure all sorts of different things about it. You can study their development, how fast they develop. Do they develop normally? Do they have proper eyes? Do they have apparent eyes? How long do they live? What happens when you test them with nasty drugs like paraquat? You can measure how fast they run around and what their eyes and legs and things do to contribute to that. You can cut them up in ways that you can't cut up humans to look for degeneration of the nervous system. You can test their metabolism. You can see what molecules are being made or not made. And you can come along and try and rescue them by feeding. None of these are exact replicates of the human situation. You don't think of them as mini humans because their genes aren't all the same. But they at least give us a chance to do things which are impossible to do with humans, or which might take a very long time to do with mice. Mice are really useless organisms for Parkinson's disease because they don't have much black pigment in the substantia nigra. And so, although they've got the same part of the brain, they're not making pigment in the same way. And this may be a serious problem for them. Okay. So in the next part of the talk, I want to discuss what happens with the gene known as Pink1. And this was first started in a whole range of labs. And the sort of research program that they undertook was to take Pink1 and look for the homologue in Drosophila, and they had identified a particular gene that hadn't been studied before, so it was called CG4523. It's an unknown gene, no previous work done on it. And so what are you going to do? Well, in Parkinson's patients, the problem is that they've got knockout of the Parkin gene and the Pink1 gene. And so you want to go and make knockout of CG4523, and that's what was done by a scheme of P element mutagenesis. And so here you see a kinase map, a map of the kinase, and you can see it's got different exons and introns, and the exons that are going to be translated are lost in the Pink1 mutant, and a different chunks lost in the Pink1 9 mutant. And they also made flies that were Pink1 rescues, Pink1 genomic rescues, by expressing the whole gene, so that they were able to test and prove that the mutations were due to the effects of Pink1 knockout, Pink1 deletion. Because when you make these mutants, one of the concerns that you always have is that you've had a second mutation somewhere else in the fly that might be giving rise to the mutations, to the observations, instead of the mutation that you're really interested in. And they observed that the flies weren't very good at flying, and they were not very good at reproducing, and they didn't lay eggs that were fertile. And so a couple of fly techniques were needed to overcome this. And what they found was that when they looked at the sperm being produced, they had got lots of holes in them. So on the left, where it says wild type, you can see that there's lots of sperm being studied, and everything looks fine. The mitochondria, the dark parts, all look nice and smooth and content, whereas in the mutant in the middle panel, most of the mitochondria are stuffed full of holes. And you can see that in panel G, where there are 4 big holes at the bottom and 1 little hole at the top. And so if you come along and you put your Pink1 gene back, so this is an artificial copy, not the copy of the fly. This is an artificial copy coming back. And so you've got both the mutant and the replacement. And you see the mitochondria all look happy, so that panel H looks just like panel F. And if you look at what happens with the flies, when they're raised, the pink ones hold their wings in a very funny way. And this is because the muscles that normally hold the wings were completely aberrant. And so in panel C, you see nice normal muscles with the organization of the thin and thick filaments, stuffed full of mitochondria. And in panel G, you see that we've got mitochondria all over the place and a great deal of swelling. The muscles are no longer nice and striated, but they've got big holes and disorganization. And in panel K, you can see that the expression of the transgene, the rescue transgene, makes for normal morphology again. So the muscles are working perfectly okay. So what was really nice about this is that 2 different labs achieved basically the same results, and they both published it in 2006 in Nature. So this picture's from Clark, and this picture's from Park and excuse me, and colleagues. And so this is about the behavior of the fly. So on the panel F, what you can see is that the proportion of flying flies is okay in the wild type, but not for B9, which is the mutant. Whereas if you express the Pink1 gene back in these flies, then about 80% of the flies will fly. And all the other things are similar. The climbing is worse, and the shape and size of the thorax is different. So two labs came up with this in exactly the same time, which was really good. And the Pink1 mutants had a whole range of other problems. They had trouble with surviving, if you gave them paraquat or rotenone or salt solutions, whatever they did, it wasn't really very good for them. And these are very relatively straightforward experiments to run. You may know about the Trend School on Drosophila neurogenetics and tech neuroscience. And here's data that was collected by students on the second one, where they had Pink1 mutant flies compared with the standard white minus controls, the wild type controls, and 100% of the white minus flies were fine after 1 day, but the Pink1 mutants, 20% of them had died, and there were only 80% alive. And similarly, ammonium chloride in the food was not good for them either. So a whole range of different things were bad. And if you warm them up to 39 degrees, then you get different recovery as well. So, of course, what you really want to know is that these effects are due to an impact on dopamine. That's what we said was important in Parkinson's disease. And so the question is, can you find any changes in the number of dopamine neurons in the brains? And so if you take an antibody and stain them, or you use expression of a green fluorescent protein, then you can count the number of dopamine neurons. And they come in little clusters. So up at the front, there's the DM neurons, and in the middle, there's the PM neurons, and so on. And there are different discrete clusters. And so you can count them up. And in the wild type, for example, in the DM, you've got about 15 neurons. And then in the B9 mutant, there's a little bit less. And the same is true for the DL1 neurons. There's a bit of a loss. And you can quantify what's going on, how much dopamine do you get at different stages. You get a loss of the dopamine in the different mutants. So that's sort of reassuring, that in young flies, the amount seems to be about the same amount of dopamine, but in older flies, there's less. So, of course, once you start thinking about the GAL4-UAS system, you can introduce any gene that takes your fancy to see what happens. And so you can say, well, what about this as a model for Parkinson's disease? If you take the human Pink1 gene, can that rescue Drosophila Pink1 null mutants? So we showed that the fly Pink1 gene worked brilliantly. Here's what happens with the human Pink1 gene, and it works just as well. So on the panel D, you can see the holes in the mitochondria, as we said before. And in panel E, when you express human Pink1, the mitochondria look fine. So really, to summarize what we've talked about, the Pink1 mutants had a whole series of defects, and somebody realized that these were exactly the same defects that were seen in the Parkin mutants. So the Parkin mutants also had funny shapes to their wings, and they had disorganized muscle fibers, enlarged mitochondria, impaired flight ability. And this fact that the wings were funny, they said, right, this might be linked to what's going on in Pink1. And so they set out to show what's called epistasis with Parkin, between Pink1 and Parkin. So the idea is that both genes might play in the same genetic pathway. So, of course, if you've got two genes that do the same sort of thing, there are a variety of different hypotheses. So Pink1 and Parkin might be friends and equals. They might do the same sorts of things. They might interact together, and you might need both of them. to work properly together. So they might be equal to each other. Or it might be that some process occurs in which you first need to do something with Pink1, and then you need to do something with Parkin. So Pink1's a kinase, so it might phosphorylate something, and once it's been phosphorylated, Parkin, which is a ubiquitin ligase, might come along and stick ubiquitin on it. So that's the scenario on the left, Pink1 first and Parkin afterwards. But the alternative might also be true, that you have Parkin first and Pink1 subsequently. So there's a subtle difference here between these three hypotheses, that you have Pink1 working and then Parkin doing its effect. You have Pink1 and Parkin acting together on something, or you have Parkin first and Pink1 second. So you can come along and you can create Parkin transgenes, and you can put those back into the Pink1 flies. So here's again some more of these pictures. In panel D, you see the same holy mitochondria. I think it's exactly the same picture as they used previously. And if you come along and you rescue with Pink1, you get nice healthy mitochondria. That's panels B and E. But panel C now shows what's happening with Parkin. And so you're getting a complete rescue of the phenomenon. So this is really a strange and wonderful part of genetics, that you take one gene and you're rescuing a defect in a second gene, so that you've got a genetic pathway with these things happening. And we also noted that the problems with the wing muscles and the wing positions, and indeed Parkin rescues this. So in panel B, we've got a disorganized flight muscle. In the middle part of it's been destroyed and come very close together. It's not nice and even, and it's full of holes on the lower bit. And although there are some holes in the fly on the right, it's much closer to the wild type. It's much better. And so the expression of Parkin is now rescuing the Pink1 problem. So the next problem is that if you come along and you take a Parkin mutant, the question is, will Pink1 rescue it? So the trouble is that what you find is that although Parkin can rescue Pink1 mutants, Pink1 cannot rescue Parkin mutants. And if you want to say what's the most important result from all the fly analysis of Parkinson's disease, I think this idea is what's really key, because it shows that you can take two genes and work out how they link together. And indeed, this has been particularly successful. It's been found in all sorts of other systems, cell culture systems, and so on in mammalian tissues, but it was first found in flies. And this whole approach has been then used to look for other genes that might be associated with Pink1 and Parkin, and there's a long list of them. And so you think, well, how do these work? What's the mechanism by which these two genes are linked together? And so here's a scheme on the right showing an reasonably up-to-date view of what's happening. So when the mitochondria are unhealthy, there's something wrong with them, then they accumulate Pink1 on the outer surface, and that recruits Parkin, and then that starts the degradation through autophagy of the mitochondria. So it comes along and gets gobbled up by an autophagosome and digested, so that you can recycle the proteins and so on that are in the mitochondria. So the steps are quite clearly that Pink1 accumulates and then Parkin gets recruited, so you can do your rescue quite satisfactorily. You can then think, well, what other sorts of genes might be involved? And one possibility is genes to do with stress responses. Once you start to have problems with your mitochondria, might other genes be active? So you can come along and you can test what happens with expression of any gene that really takes your fancy. In this case, we've got expression of FOXO and we've got expression of 4-EBP, which are stress response genes. And they rescue the locomotor behavior and the muscle morphology of Pink1 and Parkin. Panel A, you can see that the climbing ability of Parkin mutants, that's Parkin 25, is only about 40% of the possibilities, whereas controls do about 80%. And the expression of 4-EBP or the expression of FOXO rescues this behavior quite nicely and rescues their flying ability even better. So it's a nice situation. And whether you use a Parkin mutation as in A and B, or whether you use a Pink1 mutation as in C and D, then you get the rescue of the same things. And we said that one of the important things was to count the number of dopamine neurons and see what was happening. And the same stress response genes provide rescue for the not counting the number of dopamine neurons. So one of the things people are always thinking about is what's going to happen when you can find new drugs. And 4-EBP and FOXO are associated with a drug target for rapamycin. And so if you come along and provide rapamycin to the Parkin mutants, their climbing ability gets better, and the number of dopamine neurons is increased slightly. So this might be due to one of these genes that's been identified, and so we can study TOR, which is called TOR in mammals, but is called TOR with an H in flies. And you see again that you get effects. The positive effects of rapamycin are blocked by changing the amount of TOR in the fly, suggesting this is the pathway through which it works. But one of the pathways through which rapamycin was thought to work is through autophagy, and that seems to have no effect, or if anything, the opposite effect. And you can see what happens when you do the genetics or the amount of the consequences of feeding the flies with rapamycin. Then you get amounts of increased GST, glutathione S transferase, which is a detoxifying enzyme. So once you've started to identify what these genes do and how they work, you can begin to see how their mechanism of cell biology is operating. And really, I've only got a short period of time to talk to you, but there's an awful lot of papers been looking at different genetic interactions with Parkin and Pink1 and trying to see how that was done. I've just been to Liverpool a month ago to investigate, to be the examiner for a PhD, and there were 100 pages of introduction all about the role of Parkin and Pink1. So really, you know, a very exhaustive survey, and it was crammed full of references and lots and lots of information. So there are other things that we could say. So one of the problems that flies have is that they don't have any alpha-synuclein. So we've always thought that, you know, that flies made a great model of Parkinson's disease, but the main problem seems to be that they don't have any alpha-synuclein. So if you look in the brain of somebody who's died from Parkinson's disease, then you find their, particularly their substantia nigra neurons, are accumulating these Lewy bodies, which are stuffed full of alpha-synuclein protein. And this was really one of the keys to finding the first genetic forms of Parkinson's disease. It was possible to do immunocytochemistry and recognize all this alpha-synuclein being produced in the brain. Excuse me, I'm just going to have a sip of water. But this isn't really the end of the story, because alpha-synuclein is a dominant gene. So the way that you get Parkinson's disease is that you have mutations, A53T and A30P, that you only need one of these chromosomes to start giving you Parkinson's. It's early onset Parkinson's, it's very easy to recognize. It's dominant genetics. And so you could say, well, what happens if you take the alpha-synuclein gene and put it into the fly? What's the fly going to think? Is the fly going to think, I have never seen this protein before, I don't know what to do with it, I'm going to chop it up and throw it away? Or is the fly going to say, ah yes, alpha-synuclein, this is something that I know what to do with, and it will bind to particular targets and provide a marked phenotype? I suppose I wouldn't be telling you this if Feeney and Bender, in really the first Parkinson's model in flies, had looked at how alpha-synuclein worked. So in panel G here, then you can see alpha-synuclein deposits, these brown little spots in the brain of the fly, and in the muscle of the fly give you indications that alpha-synuclein is being put into something that's a bit akin to Lewy bodies. And the mutant lines do far worse than the controls. So, for example, with climbing, then whether it's the wild-type alpha-synuclein or the A53 or A30P mutants, then the climbing ability starts to fall off at about 25 days, and really they stop being able to climb well by about 30 or 40 days, whereas at 30 or 40 days the controls are still climbing quite well. There's some slight concerns about these sorts of things, that genetics wasn't quite as well done as it might have been, and it may be that there were off-target effects on serotonin neurons, but really I'm not very interested in it, because the fly doesn't have an alpha-synuclein homologue that one can detect. And so most of my work has been with LRRK2, because this is the most common cause of genetic Parkinsonism, and it's a nice typical old age sort of Parkinsonism, whereas Pink1 and Parkin and alpha-synuclein are all early onset forms. So if you have a LRRK2 mutation, then you would have Parkinson's in your 60s or 70s. And I've been looking at it both in for effects in vision and effects on movement. So this is a sort of summary of how the eyes of the flies degenerate. And what you see on the right-hand side is that the mutants in the top panel, they've got holes in their eyes and they've got dodgy mitochondria, whereas the eye of the control flies is all nice and even and there are no holes to be seen anywhere. Then working back, when you turn on a light, which is indicated by the blue bars, the red trace shows that there's almost no response in the fly eye, whereas the physiological response of the controls is nice and big and looks just the same at 28 days as it did at 3 days, whereas the controls could see controls can see perfectly well whether it's 3 days or 28 days. The mutants, the G2019S in the top panel, I can see perfectly well at 3 days, but are completely hopeless at 28 days because their eyes full of holes. And so what we suggested would happen would be that as the eye started to degenerate, you'd get autophagy and apoptosis, and anything else that you can think of that's going on as a nasty consequence. And all this is really the consequence of too much activity in the eye before any of these processes start to happen. So on day one, what you find is that the eye can see really beautifully well in the mutant line, whereas it can't see nearly so well in the controls. And so there seems to be some extra excitability or excitotoxic cascade that starts, and the eyes demand lots of ATP in the very young flies, and the demand for ATP leads to problems that the mitochondria can't keep up. By 21 days, they're really not being able to keep things going, and so you get lots of autophagy and lots of other problems on the way. So it's really work. The real question is, what's the first step down this cascade? What happens before the flies can see better? Is it a problem with calcium channels, a problem with protein synthesis? Is it a problem with making dopamine? There's a whole range of things that we're busy working on to try and see what happens there. So what we did was we did this fancy assay where we took the flies and we showed them squiggly lights, and the light intensity went up and down, and you analyzed how the fly responded. So you see a blue trace of the stimulus and a red trace representing the response to the fly, and you can see the fly does something which isn't quite the same as the signal that you're putting in, but it's sort of similar. And so you can measure this using a Fourier analysis. So there are some particular frequencies which come out because you put them in, the F1 and the F2, and that belongs to the action of the photoreceptors. And then there are ones that come in at double that signal about the on and off of the system. And then there are difference traces, F2 minus F1 and F2 plus F1, which come from the medulla. So you can begin to work out how this all fits. So here on the left is a signal, a diagram of the fly eye, or the photoreceptors in blue, and the red pigment in sort of light pink. Then underneath that's the lamina and the medulla. And so you can distinguish the identification of these components by this fancy Fourier transform. And what you find is that the mutants have a loss of vision, and that's shown by the red lines. But if you come along and feed the flies UDCA, then the mutants are rescued. So it goes from the red trace to the dark green trace. And so with this, we can begin to say, well, we can work out what's going on. And it looks if we rescue the photoreceptors, we rescue everything else as well. So, yeah. So we've tested, I think, 2 or 3 drugs in this that we've published, and we've got more on the way. So, although Parkinson's vision is very interesting, then I was really taken with this paper from Kirsten Scott's lab, because there's this beautiful picture on the right, which shows the brain of the fly in purple, and then in green there's this lovely one dopamine neuron, which has got lots of synaptic terminals in the subesophageal area of the brain. And it's really very beautiful. And this part of the brain is used in the proboscis extension response. And so the idea is that you would get dopamine modulating how hungry the fly is and controlling its response. So if you're a fly, then you're walking along on the table or something, you come across some sucrose solution that somebody's carelessly left there, and you think, oh, isn't this exciting? And you detect the sucrose solution with your legs, and then you stick out your proboscis to feed from it. So on the left at the bottom, there's a fly being offered sucrose by one of my students, and the fly sticks its leg in and then sticks its proboscis into the sugar solution. And you can see this movie if you look on my website. You can see what happens as it goes. And what we found was that the time that it took for the proboscis extension response to occur was a lot longer in the mutant flies. So the green flies are the controls and the pink flies the mutants. And you can see they're taking roughly twice as long to do their response. And you can count the number of flies that respond. And again, you find that the mutants are doing much worse than the controls. It's not quite true about all these mutants, so let's just go through it. So on the left, we've got a wild type, and then we've got another wild type. In those first two bars are in green, and then we've got flies with the expression of the conventional human LARC2. Then we've got a green bar with expression of a mutation in the GTPase domain of LARC2. Then we've got two in pink, which are mutations right next to each other in the kinase domain of LARC2. And finally, we've got one which has got both the G2019S mutation and the K1906M. So this fly has got two mutations, one of which encourages it to be kinase active and one of which prevents the binding of ATP. So the two flies that do really well at hydrolyzing ATP have a huge impact in this assay. The proportion of flies that respond is only about half the controls. And then you can come along and you can feed them with drugs, and you find, for example, that one of the kinase inhibitors that we've been testing produces a rescue and goes back to wild type levels, and so you can be able to start analyzing what's going on. And so these sorts of kinase inhibitors might be ones that would give disease-modifying potential. They would be ones which you could take and use to slow down the process of the disease rather than just to provide symptomatic therapy. So here we've managed to cover, in this hour or so, we've managed to cover the role of genes in leading to Parkinson's disease, and we've begun to ask the questions about how do these genes lead to deficits, and Pink1 and Parkin we've shown work together to affect the mitochondrial integrity and to prevent mitochondrial degradation when they're normally working. When there are mutants, the mitochondria degrade. And so then we can also ask about what other sorts of Parkinson's disease and how they work, and how you can test drugs which have got disease-modifying potentials. Okay.
Okay.